We have time for questions, but before that I should say that this morning we were talking about the VAP program training people, and Gagandeep is one of the first recipients of the training program, and so we can take pride that the VAC program has been successful. Questions? You should be here.
So thanks for a nice talk. It was quite informative for me at least. You were talking about suppressing the inflammation and having a point there that when the microbiota is less diverse or when there is the younger age, they are performing better. Now, what I learned till date, that we have a huge number of regulatory T cells, those who come up with the homing receptor goes toward intestine and guts, and they are basically required to allow the microbiota to develop. And if we plan to really go and diversify or to control the inflammation, which the T-Rex may do, what is your viewpoint here, that what could they do and what is the particular role they are doing at this stage of OPV vaccination?
And that's a wonderful time for me to tell you that the person in our group who's actually, so we collected PBMCs from these, well, first we did a description of all the cells that were there, and we had PBMCs stored from there. So the person who's actually done the T cell work is going to be here tomorrow, and you really should talk to him. He hasn't finished all of the work yet, but he does have some interesting preliminary data.
Yeah. Thank you, very nice talk. So I'm very intrigued with your ITV vaccination followed by OPV, and the difference between 6 months and 1 year, and the OPV and ITV. And so when does that mean that when you vaccinate the systemic immunity is changing over a period of time, or is it only the mucosal immunity that is changing over a period of time?
So these are kids who are primed with an oral vaccine, and then you give IPV. Then you give another dose of vaccine as a challenge dose. What you see is that the serum neutralizing antibodies go up and shedding reduces. We don't actually have a measure of secretory IgA in these kids because those assays are hard to set up for children.
Yeah, and that's Darshan.
You mentioned about azithromycin treatment, and that obviously decreased the microbial count. I understand that different species that are general there in the gut wouldn't have disappeared. But this, you also said that if there is less diversity in the microbiome, the take for the vaccine or the immune responses are better. So, in a way, the findings with antibiotic treatment and this seem a little bit contradictory to each other.
So, actually, the less diverse microbiota was not what we were expecting to find. I thought you would find a more diverse microbiota because that is meant to be an indicator of health. But we found what we found. Similarly, I would have thought that if you had older children, they might be more likely. These are kids 6 to 11 months of age, so I would have thought older children would respond better to the vaccine, but we actually found younger children within that 6-month period responding better. And in polio, it's always been that you have some amount of maternal antibodies that inhibits response, and that's been shown up to 14 weeks of age, the point at which people have studied it. But it didn't seem to hold here.
Jerry,
There is a phenomena called interference. When cells are infected with one virus, it is difficult or perhaps impossible to infect with another virus. So, is there a competition between polio and rotavirus for the same cell types in the GI tract for infection?
I think it's possible. The thing is, I thought, okay, the way I think about it, you've got so much surface area in the gut. Rotavirus targets a small proportion of the cells, which is just the mature enterocytes. Polio can actually go pretty much anywhere because most cells have CD155, right? But why, when you give rota and polio together, so nobody has done studies that have looked at shedding and correlated that with response. That's one of the things that we are planning to do. If you make an IgA response, are you also more likely to be a rotavirus vaccine shedder? And with polio, again, if you make a neutralizing antibody response, are you more likely to be a shedder? For polio, we know that. For rotavirus, it isn't quite as clear. The other thing that's really interesting is some data that came out of Bangladesh last week, which essentially shows that maybe there is interference, because even if you separate the rota and the polio vaccines by a day, you actually push up the rota responses.
So, how robust is this data on interference of rotavirus with respect to polio, immunogenicity, and vice versa?
I think it's stonking. I didn't expect it to be this much, but it is. And you can't explain this much of an effect by a 2-week age difference.
You just analyze some data from 160 trial and each trial seem to have
Well, the studies that have been done in most developing countries have shown that, but I'd be happy to discuss it with you, because what we are seeing with rotavirus vaccines is very, very low seroconversion rates. But we've been giving them with polio.
Okay, very nice talk. I had a question which partly relates to your talk, but it's more of a broader question about polio eradication. So what are your views about the change that will happen in the vaccine policy globally? As you know, the expectation is that in 3 or 4 years, or maybe even sooner than that, the live polio vaccine will not be given, and that everyone will switch to inactivate. I'd like to hear your comments on that.
I'll believe it when I see it. For polio, a lot of timelines have been pushed back, but we are probably in a better place today than we've ever been before, so I hope the momentum continues. Where rotavirus vaccines are concerned, rotavirus vaccines in developing countries seem to now be showing lower immunogenicity than efficacy. So, will increased immunogenicity also translate to increased efficacy? I can't say, because there isn't a correlation between how we measure seroconversion and clinical efficacy of the vaccine. I hope that that is the case, because currently we are at 55%. Pushing it to 75% would be fantastic.
So that's a great talk. I'm just wondering that microbiota you're talking about, do you know the specificity what type of change?
Other than in a global change, and also is it possible to have some idea about the epithelial sequencing, RNA-seq experimentation with this thing? Is it possible? Because there are possibilities that to culture the epithelial cells in the human epithelium. We culture in the lung epithelium, So that way you can look at this microbiota and then do the differential RNA-seq. That can give an idea why that is helping in the process.
So if, let me turn that, I can't answer your question, so I'll turn that around and ask you one. And that essentially is, these are viruses, these are live viruses that get in and replicate in the small bowel. What you're going to see coming out in stool is largely large bowel. You may have some epithelial cells that come from the small bowel. Now, how feasible would the kinds of studies that you described be?
It is, because you are talking about the factors can be released by this, from that system, because there must be something which is helping, and it could be a soluble factor, or that factor may go in the distal passage, and because this fascinating idea that there is a reduction of the microbiome, or that can give us an idea what, because there are no evidences that other innate system can be activated by this process, and that innate system can help to adaptive immunity. So it will be fascinating to understand that what's going on in this children. And do you have any genetic data also, those who are having this?
We have not, so this study finished like 2 months ago. So we are still analyzing samples from the study. And most of, we have blood clots from the children from which we extract DNA and store it.
Thank you, Gagan. That was wonderful. Have you looked at the data according to socioeconomic classes and basically trying to look for clean environment versus?
Yeah. So one of the things that we want to do is vaccine studies that compare children across socioeconomic classes. And we also want to compare them in different geographic locations. We have started to do some efforts towards that. I got stool from doctors' children in CMC and stool from the areas where we work. And there is a difference in carriage of gut pathogens and in stool inflammatory markers. It's very marked differences. So, one of the things we've always said is that oral vaccines would work better in children from a higher socioeconomic class. But it's not something that we have formally tested so far.
If there are no more questions, I have half a comment and half a question. My puzzle is that, with these kinds of problems, you can, if you don't get a clear-cut answer by looking at from the correlates, you are, can start exploring every possible correlation and find something or not find something. It doesn't necessarily mechanistically illuminate. It may not even be practically of value. You could go into genomes and microRNA and, microbial diversity and so on and so forth. But it seems that there are ways by which a cell biological understanding, or whether there's, interference like what Sudhanshu said, or what roles, different domains have, or what roles, the microbiota have, are feasible. So, are there cell biologists anywhere in the world looking at poliovirus entry and, rotavirus entry into cells, and who are they? Can they be brought together? Is it necessarily impactful on, vaccine programs?
Well, poliovirus entry is something that lots of people have studied. There's a much smaller group that has worked on rotavirus cell entry, but nobody's actually approached it as a way of understanding why this happens, the lower seroconversion rates.
And so a related point was, historically, in terms of epidemiology, do we have data from OPV administration in different societies and how that response has morphed with time? Does that give us any indication?
So it's very interesting that I said seroconversion rates in the studies that were done in the field in India seem to show very low serum neutralizing titers. But if you look at contemporaneous trials, last 3-4 years or so, seroconversion rates are looking pretty good. It's much higher than was reported 20 or 30 years ago. And this is not in Tamil Nadu, which is a different part of the world, but in UP.
Good. I am standing next to stimulus, but we'll soon go there. Thank you very, very much, Jerry. Very stimulating and exciting talk. We have some formalities to complete, very important ones. Jyoti has a red box and she's pointing to Cherry, so I must do something with that, I think. Here you are. Thank you very much. There's still graduation. Oh my God, something.
You have to go to the
Oh, this way, sorry. You have to hold this.
Flowers this way?
Okay, hold on. And now there's something equally important. Gray has got a surprise announcement or two. He's carrying a bag, so.
That's right.
I'm
holding the bag. Today there were many discussions about the history of this program, what it had accomplished over time, and what an exciting future it has. One of the both wonderful things about the program is that there have been so many brilliant people and hard-working people and visionary people involved in this program. You heard two of them mentioned in the naming of the lecture that Cherry delivered. Professor Ramalingaswamy, one of the great public health and science thinkers of history in India, and Professor Robbins, who was along with Professor Ramalingaswamy, were the two founders, really, of this organization. But they were followed very quickly by people that made a difference by working very hard, by being very knowledgeable, and by solving every problem that presented itself for a new program, a growing program, and now a very mature program like the Vaccine Action Program. I mentioned in my talk earlier today that the program had really been important in 2 generations of scientists in the United States and in India. It's also been very important to 2 generations of government leaders in the United States and India. And this year, we have lost 2, although we haven't really lost them yet. We have lost to the formal engagement with the Vaccine Action Program 2 of our most important leaders. So tonight, I'd like to recognize with all of you the very important contribution made to this program by Dr. Carol Heilman and by Dr. T.S. Rao. I'm so glad to see that T.S. is still here, providing his advice and wisdom throughout the day today, and hopefully throughout the day tomorrow, and throughout the future as we move ahead. Although you've retired from your formal responsibilities and no doubt spend a lot more time at Goa and other places than you used to, I think we're still going to need to rely on you for your advice and your wisdom. So we have a little something to express the appreciation both of the National Institutes of Health and the Department of Biotechnology and the Indian Council of Medical Research. And of course, flowers. Thanks, it's a surprise Make sure I've got, yes, this is the right one.
Thank you very much.
We should, Greg, we should take a photograph of Soumya. and
Oh, that's
Carol.
perfect. Good. Here, come over here. Well, you need your.
It's my buddy. Yeah.
I was going to do them separately.
Yeah.
There we go. And I'll give you yours too.
Thank you. Thank you. It's a great.
There you go, Carol.
Oh, thank you so much. Thank you so much.
Thank you.
And Carol, like TS, we're going to rely on you to return frequently to provide your wisdom and make sure we continue on the right course. Thank you all.
Thank you.